Good afternoon, everyone. Welcome back. Thank you for joining us. We're glad that you're here. I'm Steve,
And I'm Scott.
and together we're Backyard Musings, the science and technology channel.
Yep, and we're talking about another health issue, kind of like we talked about this morning. This has to do with trying to survive a stroke.
Yeah.
I mean, this is important stuff because strokes they're like the silent killer, right?
Yep.
You never know.
Never know.
Yep.
Scientists have discovered that transplanted stem cell derived brain cells may do far more than simply survive after a stroke. A stem cell treatment helped mice recover from strokes by rebuilding damaged brain connections, restoring blood vessels, and improving movement, according to new research from the University of Zurich and the University of Southern California. The findings raise hopes that future therapies could one day repair stroke damage that is currently considered permanent. Would you like to know more?
Okay, strokes remain one of the world's leading cause of long-term disability. When blood flow to part of the brain is cut off, oxygen-starved cells die within minutes. Unlike skin or bone, the brain has only a limited ability to replace lost tissue, leaving many survivors with lifelong paralysis, speech problems, or memory loss.
Scientists have spent years searching for ways to help the brain rebuild itself. In the new study, researchers used neural progenitor cells, early-stage cells capable of developing into different types of brain tissue. The cells were created from induced pluripotent stem cells, which are adult human cells reprogrammed into a stem cell-like state. Say that fast 3 times.
I can't say it once. The team transplanted these cells into the brains of mice 1 week after stroke. What did they cause this mouse to have?
They caused humanized mice to have a stroke.
Okay, where's PETA? That timing turned out to be critical. Earlier transplants survived poorly because the injured brain was still overwhelmed by inflammation and toxic chemical signals. Waiting several days allowed conditions to stabilize enough for the transplanted cells to take hold. What happened next surprised the researchers.
Over 5 weeks, the transplanted cells survived, spread through nearby brain tissue, and matured mostly into functioning neurons. Many became GABA-ergic neurons, specialized inhibitory brain cells that help regulate neural activity and are heavily depleted after stroke. These cells are essential for balancing brain signaling, preventing excessive excitation, and coordinating movement.
The grafted neurons did not simply exist alongside the damaged brain cell or brain tissue. Evidence suggested they actively communicated with surrounding cells through molecular signaling systems linked to neural growth, synapse formation, synapse. Okay, synapse formation and tissue repair. The researchers identified several major pathways involved in this crosstalk, including neurexin, neuregulin, NCAM or NCAM?
NCAM, yeah.
And SLIT or SLIT signaling, all of which are associated with rebuilding neural networks and guiding axons to reconnect.
That was very technical, Scott. Very good job.
And once again, I knew nothing about what I just said. So
the stem cell treatment also appeared to trigger a broader healing response across the injured brain. Mice receiving the transplants developed significantly more blood vessels near the stroke site, improving circulation in damaged tissue. The treatment also reduced inflammatory activity and strengthened the blood-brain barrier, the protective lining that normally prevents harmful substances in the bloodstream from leaking into the brain. Damage to this barrier is a major contributor to swelling and further injury after stroke.
Researchers additionally observed increased growth of nerve fibers along the damaged region. Some transplanted neurons extended long projections into areas linked to movement and sensory control, suggesting the new cells may have started integrating into existing brain circuits. So this is like rebuilding the brain.
Yeah, yeah, and it's doing itself. These stem cells are doing it.
Yeah.
To measure whether these biological changes translated into real recovery, the scientists used AI-assisted motion tracking systems that analyzed how the mice walked. Stroke typically disrupts coordination, paw placement, and gait timing. Mice treated with stem cells gradually regain smoother movement and perform better on balance and fine motor tasks than untreated animals.
The recovery was especially noticeable weeks after transplantation, indicating the therapy may support long-term repair rather than providing only short-lived benefits. Quote, our findings show that neural stem cells not only form new neurons, but also induce other regeneration processes. And that's according to Christian Tackenberg of the University of Zurich's Institute for Regenerative Medicine.
The study also addressed one of the biggest concerns surrounding stem cell therapies: safety. The neural progenitor cells were produced using animal-free methods designed for future clinical compatibility. Researchers are now developing built-in safety switches that could shut down transplanted cells if abnormal growth occurs.
Oh, okay. Another major goal is making treatment less invasive. In the current experiments, cells were implanted directly into the brain. Scientists are now exploring whether stem cells could eventually be delivered through blood vessels instead, similar to minimally invasive stroke procedures already used in hospitals.
Several types of stem cell therapies have already reached early human trials for neurological diseases, including Parkinson's disease. Stroke may become one of the next major targets. Important hurdles remain. Experiments were performed on genetically modified mice whose immune systems would not reject human cells, and researchers have not yet proven that the transplanted neurons fully integrate into human-like brain networks over the long term.
Quote, we need to minimize risks and simplify a potential application in humans. That's according to Tackenberg. Going on with the quote, stroke could be one of the next diseases for which a clinical trial becomes possible. Again, if your family is susceptible to that, and you show signs of it, sign me up, right?
Sign me up. What do you got to lose?
I want to be at the front of the line. I'll sign a waiver. So, good story. Two good stories today.
Yeah. All right, folks. Thanks for joining us. We'll catch you in the morning.
Take care, everyone. Thanks. Have a great evening.